Namaste and pranam everyone. We are again here for our monthly KSAS Talk series. And as you know that every month we come with a very esteemed scholar to discuss the various topics on humanities, science and other disciplines. So we have with us today Dr. Bittu Kaveri Rajaraman, who is an associate professor of biology at Ashoka University. He received a PhD from Harvard University in neuroscience and then went on to become a DST-Kothari postdoctoral fellow at the Center of Ecological Sciences, Indian Institute of Science, and then an Inspire faculty fellow at the Central University of Hyderabad. He works on the evolution of neural and behavioral system of communication, the neuroethology of temporal pattern recognition in insects, and the quantitative and economic cognition more broadly in zebrafish, dog, and humans. So we welcome you, sir. And today's topic is how do neural systems evolve? So now the dais is yours, please.
Great. Thank you very much. Thank you for inviting me to speak on this platform. So I'll speak for about 45 minutes, if that's okay, and then pause for questions.
Yes,
Does
yes.
that sound right? Okay. All right. So I work on the broader question of how neural systems evolve. I know many of you here would perhaps largely be interested in neural and cognitive systems. And I'll explain why I work on the particular systems I work on to understand how neural systems evolve.
Thank you.
So as I said, there are two broad questions that I work on. There is communication systems, how communication systems evolve, the neural systems underlying them, and how quantity perception or quantitative cognition evolve. And the simple way of explaining why I care about these two things or what ties these two things is, what do you do when you're in first standard or second standard in school? You're basically learning maths and you're learning, some language. So that's kind of what I work in my lab: communication as in language systems and maths as in quantity cognition, right? So that's a simple way of remembering kind of the kind of cognitive tasks I'm interested in. And we understand something about how the neural systems that produce communication behavior in our brains work. But it's somewhat limited because our only, we have limited ethical, frameworks within which data from human brains can be collected with high single cell resolution. And, very few other animals share the same kind of communication that we do. Of course, our insight into their communication is also limited by how little we understand of animal communication. And there are good models for how songs are learned, how communication is learned, and so on, largely from bird and monkey models. But all of these represent learned communication systems. That's a hard problem because as obviously neural systems learn, they also change. So if you want to look at the neural system underlying communication, it will change as learning takes place. And then, finding, it's basically a more complex system to understand than if you have a system like the insects I work on. These also produce calls ranging from simple to more complicated sounds, but they're much simpler. Both the communication signals themselves are much simpler, and the neural systems are simpler. Now, I don't want to say simple in the sense of being, necessarily computationally less ingenious. But I just mean that they're smaller neural systems. And when there are smaller neural systems, you need to keep track of fewer numbers of neurons before you can kind of understand what they're doing together. So, and of course, C. elegans is the even simpler system, where you have really good single cell insight, but we don't have a lot of, I mean, C. elegans acoustic communication, right, is not a system we can work on. So in some sense, I would posit that this is a system that's a nice, simple system to look at communication. Now, the other thing that makes these insects, and which insects am I talking about? If you have stepped into any village or any forest in India, you will have heard at night krr, 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 right? That kind of sound. And so that kind of sound is, what you will, those are the kinds of sounds produced by the animals I work on. They're called crickets and bush crickets. And they are on the left side of this slide as you're looking at the slide. So these are, they belong to the same order as, what's called, they're called Encephora. And they also belong to Orthoptera, the order that also includes grasshoppers and locusts. So the way you can kind of tell them apart is that, first of all, grasshoppers largely call during the day. They have short antennae, the kind of the size of their faces. In the old days, they were called short-horned grasshoppers. Of course, that's not a horn, but the antenna. And crickets and bush crickets have these long antennae that are almost as long as their body, as you can kind of see in the slide. And grasshoppers and locusts are largely calling in the day. Crickets, bush crickets are the ones that are calling at night. So raat ka jo chorus hai, that is largely crickets and bush crickets, the ones that I work on. But both groups produce sound, right? But they produce sound in totally different ways from one another, indicating that sound may have evolved independently, both the production of sound and the adaptation of organs for listening to sound in these two lineages. So grasshoppers and locusts produce sound by rubbing their legs together, and they hear using a ear which is actually on their belly, on their antennal area, abdominal area. And the abdomen has a modified mechanoreceptor, which would normally have felt touch. And of course, a ear is just is something that detects very small vibrations. So it's an exceptionally sensitive mechanoreceptor. And in the case of grasshoppers and locusts, the mechanoreceptor has evolved, from a mechanoreceptor on the abdomen. And in the case of crickets and bush crickets, a modified mechanoreceptor on the front leg of the animal has become the ear. And they produce sound by rubbing their wings together. Okay, so very different way of producing sound and of hearing sound. And then even within crickets and bush crickets, there are many groups. In fact, many of the groups don't hear or call. And there are these two groups, ground crickets and bush crickets, which produce sound in similar ways. Both rub their wings together, but there are some differences. Ground crickets rub the right wing over the left wing, and they produce low frequency calls with pure tones. So that krr, krr that you hear is often a ground cricket. You can hear it calling from the ground. But usi time pe, if you hear from the trees, you will hear sounds like, right? Those are bush cricket sounds that are coming from the trees, and they produce high frequency broadband kacha 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 sounding noises, and they rub the left wing over the right wing. So one of the open questions is, in this area is also, did these evolve separately from each other also? And if so, did all the intermediate lineages who don't produce or hear sound, did they lose the ability secondarily from a common ancestor that produced sound, or is it the other way around? Did the common ancestor not produce sound, and did it independently evolve in these two lineages, but using a similar kind of, with a modification of the same kinds of organs? Is it convergent evolution essentially? Okay, so now I'm going to play to you the song of a cricket, which hopefully all of you will have heard. It's found all over India. It's one of the loudest crickets. It's not a ground cricket, but it's a bush cricket. And this is the monsoon. So as monsoon, once the water falls, all the eggs will hatch. And so abhi, they might be really tiny babies, about this size, the size of, tiny, tiny babies. But as they attain adulthood, they start to, their wings develop. And once their wings are developed, they start to produce sounds like this. I hope all of you can hear this. Yeah, so this is an animal called chirper, and it's from a genus called Mycopoda. And part of our work focuses, actually much of the work, this talk focuses on this Mycopoda. So in most of India, you will hear only this Mycopoda. I mean, I have been to Punjab, Haryana, UP, Maharashtra, Madhya Pradesh, All of these are north to south. I have not traveled east enough to hear, and I've these animals even in Kolkata, Odisha. So that full, like almost of India, you hear this animal. Some variations in the call, krr, 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 kind of pattern, which is called chirping. So even within the krr, each of those small, those are what are called pulses. And then the whole thing is called a chirp. Krr. And then there's a pause or an interchirp interval. Krr, krr, and so on. Okay. So I became interested in this animal because only in the tropical rainforests of two parts of India. One is the tropical subtropical rainforest found in Western Ghats, and the other in the area of the Northeast. Our particular work happened to be in a place called Nokrek in Meghalaya. And in the Western Ghats, past work has already found that there are up to 5 morphs of Mycopoda in Western Ghats, which all produce different song patterns. That is to say, they all look like this animal, but the pattern of the krr krr varies between them. And we tried to see if similarly these types are only found in the Western Ghats, or are they found in other subtropical rainforests? Because basically what we cared about is to look at the neural circuits underlying the production of such sounds. And in Nokrek village, we actually found a wide variety of Mycopoda. Now, all of these, you might think all of these look different from each other. They must be different species. But it turns out, if you actually look at all of them, only the coloring is actually superficial. So it looks just like with human races. People think that there are deep genomic or biological differences between human races, but there are not. We are a remarkably alike population genomically, with these superficial differences of coloring. And the same thing is true for Mycopoda. So green Mycopoda ho, light brown ho, dark brown ho, they are all, it looks like the same genus. But within even each call morph, you will see that they can produce one of many different call types. Now, let me explain what some of these call types are. The standard chirper जो मैंने आपको दिखाया वो तो है ही. Okay, it sounds slightly different in Northeast. Then there is this animal where the duration of the chirp varies. Sometimes it goes prr, prr, sometimes it's prr, prr. So these variable length chirps. That's 2 call types. Third call type is this one, which has regular calls, but they seem to be much shorter. Then you have what we call a true triller, which is just continuously calling. But you can hear that there is a cyclic nature to the song. Right? And you can see that in the spectrogram, which is what you see below. What you see is this, just the profile, the outline of when the call is on and off. And then finally, we have the most complex caller of all. It's going to take me a full minute to play this song to you. So you see it starts with short chirps, and then these will start to lengthen. Lengthen even more and become a trill. And then after it becomes a complete trill, it will become louder and louder. It takes a while to get there. But this whole complicated thing is one animal which looks like all the other call types. One subset of the callers are like this. Now it's a full trill. No break.
Are you playing something?
Oh, can you not
Excuse me.
hear the sound?
No, I didn't.
Oh, that's terrible. I've been playing all of these calls and hoping you could hear them. Maybe I do in my, I'm just going to quickly reshare and see if while sharing, I need to share sound. Okay, now I'm just going to try this again. Okay, I'm sorry, I'm going to have to play this one very quickly and then I won't play the other call types. Can you all hear the sound? Yes, okay. This is the chopper. Okay, then you have various choppers, these with these long but slightly random chirp durations. Then you have a true triller, which sounds like this. So you hear that there's a circular kind of quality to it. It's continuously trilling, but the quality of the sound is good. Right? And finally, this is the complex call. Oops, sorry. Okay, so start with small chirp. And then I'm going to describe it because we don't have enough time to play it all over again. But it becomes longer and longer and longer. It takes a minute for this to become a complete trill. And then it goes back to being So it's a 1 minute long repeating song, which is complicated. We just call it complex. To indicate that it's not one type of call. It's not repeated or repeated. So all of these different callers and call types look similar, morphologically similar, except for these superficial differences in terms of whether are the animals green or blue, brown, sorry. You can see the genitalic structure is basically conserved across them. Sometimes these animals which look the same are actually different species, but then you'll see something show up either in the wing structure, which produces the type of song. You know, there's a row of teeth under each wing, which the other wing strikes again. And in the bottom row of this slide, you can see the CT scan of those rows of teeth. And they are also remarkably similar across the animals, which also checks out because the frequency of the song produced by all of these types is not identical, but largely kind of similar and overlapping. So they morphologically overlap, but the call temporal pattern is largely distinct. And we were able to get single gene data to try and place all of these animals in a sort of chronology to get some kind of sense of how they might have evolved from one another. So here you can see a phylogenetic tree. And all that this tells us, single gene data is very limited in terms of how much you can infer from it. So we can't really infer or confirm species identity. This is with respect to cytochrome oxidase gene. But the only data we have for Mycopoda other than our species in this area are single gene data. So the only phylogeny we can construct, not just with our data, but we're looking at these Mycopoda from China and Japan and all the other places where these data have been collected, we only have single gene data available. So to try and create some sense of the biogeographic history, this is kind of a first step. And we see that the simple chirper, the complex call, and so on, clustered together with all the other known Mycopoda of India. But only those trilling calls seem to cluster a little closer to the Japanese and the Chinese versions. Japanese, as you can see, Nipponensis, and Haiyanensis is the Chinese caller. So what do we do then? We bring these animals back to the lab and we try to look at the neural circuit that produces this type call. Now, Stefan Schoenig, whom you can see here, is also someone we worked with. He has done this kind of work with ground crickets, Gryllus bimaculatus. And, they found that if you look at the animal's nervous system, which is laid out on the left part of the slide, brain on top, abdominal, tail abdominal section below. All the A1, A2, A3, A4, 5, 6 is abdominal ganglia, that is groups of neurons. Then there's one in the brain is here. They have one right under the esophageal ganglion, right? Subesophageal is called right here, basically the equivalent of under their jaw. And then there's the first thoracic segment, second and third. And then there are the abdominal segments. So 3 thoracic segments, just like ants, from which the 3 pairs of legs come out. And the wings are also attached there. So you would think they found that pink arrow points to the spot where they have found the neuron in the brain that gives the command, ki ab gana gao. Okay, so that sends a signal. You would think, ki wo signal, it has to go to the wing. So somewhere between the brain and the wing would be the circuitry, the neural circuitry that is responsible for producing this pattern of song. It turns out that is wrong. So what Stefan Schoenig and their lab, Bertolt Hedwig's lab, is that the abdominal segments, these may not zaruri are involved in song production, but they found as they cut off abdominal segments that the song pattern was completely disrupted. Okay. So they concluded that the abdominal segments contribute to the patterning of the song. And we asked the same question with our bush cricket mycopoda because we want to look at how, if you have these 5 different callers, what are the differences in the neurons that are responsible for producing those sounds and that are producing these different communication patterns. Eventually, we all want to understand complex communication. And this is a beautiful system where you have 5 animals, all of the same genus, looking alike, but producing complexity. By that, I mean, singing 5 songs. One animal is only singing 1 of 5 songs, and it sings that one song lifelong, all sitting next to each other on the same bushes in the same. So it's an interesting question how these differences have evolved. Are they mutation driven? There are many questions that we have. So we did different kinds of cuts. A shows you just completely cut the neural cord under the abdominal segments. You can cut at different stages of the abdominal segments. We also have C, which is our controls, where we do not cut the neurons, but the rest of the surgery we do. So you have surgery controls. Basically, if after you cut up an animal and cut the nerve, if you don't have sham controls, then a reviewer can correctly ask this question, how do you know you did not just traumatize the animals by subjecting them to a surgery where you cut into the body? So the sham controls, we do all the rest of the surgery, same process, except we don't actually cut the neurons. So it's a sham cut of the skin, and then we seal it up in the same way. So with all these animals, after you make the nervous system is right under the skin. So it's like making a tiny superficial cut and the area to desensitize it. You make a superficial cut in the skin right underneath other nerves. So it's like the skin is the nerve. So you make a cut of nerves like this, and they literally look like two. You make a cut in the nerve. If experimental condition, and if it's not the experimental condition, you only made above it, and then you put Vaseline on the Vaseline kind of seals in the lip, and then the animal is able to heal. So heal करने के बाद क्या होता है? Well, the good news for our bush crickets is that after healing, they sound the same. They call, so it means they recover also. मतलब खुश हो गए फिर से. They are able. It's not like जीने का मतलब the drive to live has not been removed. And you can I want to show you what it looks like over here, so you can see the animal. I'm just going to overlay the neural system. This is the animal, remember, with its wings would be at the back, and it's on its back, so face is up. Okay, you can kind of see the mouth parts on the left. And you can see this gleaming portion, which is where we've cut the connective. Can you see a little cut in the animal? This is just to show you what the cut looks like. So it's the whole thing is transparent. You can barely see anything. But for some of them, we have taken pictures with more clarity of the nerve cut itself. But I just want to overlay this so that you can see where we have cut. Right? You can see thoracic segment 1, 2, and 3, and the abdominal segments below. So once you change it, if you measure the parameters of the song, that is how long does the krr last, how long does the gap between the krr last, all of these seem to be the same except for one parameter, which is the krr, the interpulse interval seems to have changed. Otherwise, there seems to be no difference whether you cut between the thorax and the abdomen. That is the first thoracic abdominal fuse segment and the next abdominal segment between what's called T3 and A3, or between the third and fourth abdominal segment, A3, A4. If you cut just one of the two paired nerves instead of the other, still no difference. They all look exactly the same as sham controls. So this leads us to conclude that in this subtropical region, we have 5 call types, which are what are called cryptic, which means they look alike. Syntopic, which means they're found in the same, and sympatric, which means they're not just found in the same general kind of geographical area, but in the same habitat as one another. These are similar in fine morphology and call spectrum, but the temporal patterns sharply differed. And all we know right now is that the song patterning to circuit lies, the way you would commonsensically assume, between the brain and the abdominal segments. It does not involve the abdominal segments. But this is a difference from what we know for ground crickets. And it suggests that even though these animals may have had a common evolutionary origin, or, I mean, that we don't have enough data to conclude whether they have a common evolutionary origin or whether it's convergent evolution that happened across two different kinds of lineages. But what we do know is that the neurons underlying the song production circuitry seem to be the neural circuit seems to be quite different. Now, we also don't know if this is all bush crickets versus all ground crickets. So now one of the things we're doing is looking at more bush crickets to see whether this is a common theme across bush crickets or is it just Mycopoda, and so on and so forth. Our lab does a little bit of other work. I decided to focus on this story for today, but I'll just give you a glimpse of the other things we do. Have an opportunity for another talk. I'm happy to specify more. So one of the things that we do when we're looking at this kind of thing, this is just our setup in the lab. There you can kind of see on a hook electrode the neuron producing. If you can kind of see this little thread which is very, very transparent to the nerve. That's the setup that we use to do recordings from that paired nerves. And this is our experimental setup. And to all of the work I showed you so far, we are also staining using dextran dyes to stain the connectivity between different parts of the nervous system. We've also had interesting successes which we did not anticipate when we started this kind of work. For example, with another bush cricket who we're looking at called Hexacentris, it turned out that the kinds of Hexacentris we found, not just in Meghalaya, but even Ashoka University campus, which is in Haryana, which is, people are always looking for new species, exotic forests, including us. But turns out one of the species we had just calling in Haryana next to our camp, a new species. So we named it after the campus. We called it Hexacentris Ashoka. And we found another one thoda dur, near the campus, but just outside in the village. So गांव में सब लोग इन animals को टिड्डे बोलते हैं. So we called it Hexacentris tidde to honor the state of Haryana where we found these animals. And then in Meghalaya also we found this Hexacentris, very different species called Hexacentris khasiensis. Again, these are three animals which look kind of superficially alike, but unlike Mycopoda, when you look at the fine morphology, they are distinct enough to be three different species. And they use very, very different songs also. So we were able to describe these different species and hear the call. This is the call of Hexacentris tidde. Many people hear this, and I hope you can hear. Many people hear it and think it's like some, high electricity tar jab buzz karta hai na. So even when we heard it, we first thought it sounds some mechanical noise. Because it sounds like that. And then we realized, no, it's coming from the bushes. It's not coming from the wires. So that was Hexacentris tidde for you. This is Hexacentris Ashoka. Okay, very different call as you can hear, but the same quality of metallic sound. And the complicated one. It goes. Okay. These are the parts of the wound that we call it, and you can see underside of the wound has these on it, which is what the other hard parts strike the plectrum strikes, and it makes the whole wing resonance. After guitar string sitting behind it, the whole back of the guitar vibrates in resonance with the plucked string. The same way, these animals, right, show this kind of resonance, right, between this part that is struck and the entire structure of the wing. And these are their bodily morphological differences and their very distinct acoustic calls. And again, we were able to do some molecular phylogeny with these, showing how well separated the Ashoka morph and the Khasiensis morph are compared to the other morphs. Okay, so how do songs diverge and evolve? Well, we know that some of these will be driven by mutations, some mutation that changes either the frequency of the song by changing the spacing of the teeth on those wings or the structure of the wing, change the frequency of the call. Any change the neural circuitry underlying it, change the temporal pattern, change the listener's preference for what kind of call you even recognize as your own species. That is sexual selection, right? So in sexual selection, we have understood a lot about the peacock's tail and why is the peacock's, what makes the peacock's tail so large. But what makes the female peacock be interested? What changes in the neural structure makes female peacocks be interested in an ornamented tail? That kind of neural basis of sexual selection has not really been looked at or not been investigated to the same degree. And that's something that I find very interesting. And finally, we have longer term questions about whether the concerted evolution between sound production and sound reception, because, if these don't co-evolve, so if one animal starts producing a sound that is not recognized by other animals as ye mera species ka sound hai, then it's not going to be reproductively successful. Likewise, if some animals start to prefer songs that are not really being produced by the species, they may not be reproductively successful either. So do sound production and sound reception co-evolve just because of the selection pressure imposed by the success of the communication? Or are there any genes that link both of these or neural circuitry that links them? These are some of the questions we're interested in. We want to look at various aspects of the neuroscience of listening as well. In fact, that's how I started getting interested in this work, which I started with Professor Rohini Balakrishnan during my postdoc at the Center for Ecological Sciences at IISc. And finally, we want to look at, what are the selective forces shaping the evolution of neural systems and how song might drive speciation. Finally, we do a little bit of work in the lab on quantitative cognition as well. This is largely work we do in the zebrafish, which, like C. elegans, offer the advantage that you can look at the whole brain in some detail, with single neuron resolution. And we look at how fish make decisions between groups of zebrafish of different sizes. We use machine learning algorithms to track the fish. And we have a bunch of data from that, which I'm not showing you in this talk, just so that we focus on one story at a time. These are the members of my lab. In the middle, you see Arani, who did all of the fieldwork in Nokrek, along with Riban on the left, who is a student from Meghalaya who wanted to study the animals around his home state. Vivek on the bottom right, who's done all the work on the neural system, along with Sayan, who's a student, and my collaborators Ranjana on the right and her student Munal. Guru, who set up labs recording systems. Vikrant, Shweta, Anindo, who works on the genes and sound production and reception, and Jishnu, and all of our field assistants you can see below. These are the grad students, who work on the fish song production system. And these are all of my funding acknowledgements of the Meghalaya Forest Department and the community around us at Ashoka. Thank you very much. I'll stop, and I'm hoping to take lots of questions.
Thank you, Dr. Rajaraman. It was quite interesting. My colleague, Dr. Abhinandan, was saying that we are hearing a very new perspective of evolution.
Right.
Others. So now the session is open for, question and answer. So one by one, I request you to please ask if you have any question, if you have any comments, so you can ask.
Okay. Thank you. Very, very informative and very, very different kind of perspective. And first time I'm able to know that the cricket sounds are so much of different frequencies also. My question is, since you said that the neuron is not between the wing and the thoracic, right?
Yeah.
So what means is there the neurocircuitry is similar to what we have as a spinal cord, and then we have other connections to do our movement, and this does the same uh, f- uh, anatomy is there for the cricket.
Good question. So, in fact, it's an excellent question with a really interesting answer. So it turns out the cricket ear is very similar to our ear in the sense the inner ear. So first of all, they have the ears on the front legs. In fact, maybe I can show you some interesting slides on this. The ears are, yeah, I should. Okay, so if you can see, it's one of the animals that I've worked on. That's its called. You can see here a CT scan of that I did of the ears. So you can see that the ears are on the front part of the front leg, right? And there's a reason for that. If anyone wants to know, it makes it easier for you to judge direction when your ears are further apart. But the direction of the incoming call. But you can see, first of all, that on that elbow, it looks like 2 ears. You know? There's like 2 coverings, the way that we have one covering per ear. They have like 2. And then behind those, they have 2 tympana. So not one eardrum, but 2 eardrums. And you can kind of see that in that CT scan of the ear on the top right of the screen. So it's a different kind of ear, but the inner ear, that is the part of our ears and inner ear where like the primary auditory receptors and bounded by a kind of by the covering of the inner ear. They have the exact same thing. And you've seen our cochlea, which is curled up. They have something very similar called the crista acoustica, which is a membrane which, like the cochlear membrane, starts out thick and floppy at one end and ends up stiff and thin on the other end. So the structure of their inner ear is remarkably similar to our ear. And that is definitely a case of convergent evolution. Because most people don't have that kind of ear. That part of their neural systems is very similar to ours. Of it is not. In case we know completely for sure that the patterning of sound happens inside our brains and is just sent to the vocal cords. And there's no question that, if you get paralyzed, so as people who get paralyzed, say, because of a spinal injury, they typically will not lose their ability to talk because the communication circuit is between the head and the neck. But in the case of these animals, somehow, can you imagine? Well, first, of course, insects have more distributed brains throughout their bodies than we do. It's not all concentrated at one end as much. But yeah, this would be the equivalent of our song, our call production starting here, then the circuitry going down the spinal cord and coming back up to influence the vocal cords in the tract. So that would be the equivalent. But excellent question.
Okay, and then second part related to this is so you touch briefly on the cognition part. So they also their sound also evolve over the period of time with the you use the term co-evolution. So in
Yes.
during that so their sound means I don't know how old these animals are. I'm supposing uh, um, assuming that they are almost 50-60 billion year ago, a year ago. They started from that period of time. The sound evolution is connected to their cognition or not?
Yeah, so from the beginning, we think it would be connected to their cognition. And good question. Yes. So as you say, you're in the right ballpark when you say 50, 60 million years ago. So not even, but it's even older than that, little bit. So it's, we are only sort of as mammals also, we only date back to about 60 million years ago, but they date back to the Jurassic period. And at least they may date back even earlier, but fossils have been found of these animals in the Jurassic with the wing structure preserved. And based on what we know of modern insect wings and those insect wings, we can even reconstruct the kind of sound they would have been made when they struck against each other. So this kind of very cool paleobiology gives us the identity of these insects as the first acoustically communicating animal we know of on land, and which is very cool. So yeah, Jurassic period. So about 100 million years ago is when we think we know that they had a song production system at that stage. And so they've been evolving for a very long time.
Yeah, so you know, last time also I invited you for our symposium.
Yeah.
Because of the exam, I think you were not able to join us. But next time we will have, and please have your dates available for that. But in that, we want to also that symposium is to discuss about the different cognition system and how they involve, and how they are connected to the human cognition system. Means they must be a part of the co-evolution of all the animal, insects, and plants together to develop intelligent system like human. Probably we are not the intelligent system, but still we, in our definition, we are the intelligent system. So that discussion, how you think that the insect evolutions, the brain evolution of insects is connected to human evolution of brain?
So excellent question. We have a lot of evidence of the co-evolution of these insects with mammals. Specifically, so,
Dr. Raj Gopalan,
because I think can
see,
you all
yeah.
hear me? I feel like my belly being should be part of the.
Yeah,
Okay,
I think.
so I'll continue. So yeah. So these animals definitely have co-evolved with bat sounds. So we can see very clearly that bats have these animals have co-evolved because they, as I said, ground crickets call exactly between 4 and 5 kilohertz. And that is exactly the bandwidth of sound where as in yours, my, dogs, cats, and bats, all of us have the worst directional abilities at that frequency range. So, we judge the direction of incoming sounds using two different kinds of computationals, computational tools. One is a sound arriving at this year before it arrives at this year, so timing difference. So basically, we know this from work on owls that when sound reaches this year versus this year, it then travels through circuitry. And so then there are coincidence detectors along the way. So if the input from these two neurons reaches at the same time over here, that suggests that the sound arrived at both ears at the same time. So the sound source is either in front of me or behind me, where it reached both my ears at the same time. But if it arrives here first and it arrives here later because the sound is on that side, so it comes here first to this here and starts moving down these neurons and reaches this ear later and then starts moving. Meet over here, like the two signals will come into this neurons. And then a coincidence detector firing here tells me that the sound source is somewhere there. So that's one way of computing. And then the other way of computing it is just loudness. Is it louder in this ear versus that ear? So, but we use different computations at different frequency ranges. And 4 to 5 kilohertz is the region between where both of these mechanisms function poorly. So it's where we are worst at hunting crickets. And all ground crickets call at this frequency range. So they have clearly co-evolved to not be heard by us. And if you look at the largest neuron in the cricket system, the neuron that is often the easiest to record from, these are neurons that are, especially in the bush cricket system, it's a neuron that detects bat sound and immediately leads to an escape response. So they are listening to us meaning mammals, especially bats. And we're listening to them. They've evolved to call at frequencies where we are bad at finding them. So in fact, one of the fun things that I ask people is, have you ever tried to catch a cricket? And of course, anytime a student comes to hunt crickets for the first time, the first thing you realize is you're moving towards the cricket. It sounds like it's calling here, but wahan paas mein jaake you realize, wait, it's not here, it's somewhere else. So, and many people who have chased crickets around their house, you'll hear the cricket. It's very loud sometimes if it's calling at night, if it gets inside the house. They can produce 100 decibel at source. It's very, very loud. It's deafening actually for your ears. So, and if they happen to be in a part of the room, which is like, walls and floor, concrete floor, then that amplifies the sound. It echoes and it can be very loud. So people often try to chase them out of the house. And chase karne ke liye bhi you can't find them, because you go one direction, then you realize the sound's coming from behind me. And people assume the cricket is jumping, and that's why the sound moves. But usually crickets don't jump. They rely on being camouflaged or being hidden, and they don't usually move around. If they were to move around, we'd find them much more easily. So especially in homes. So there is very much coevolution of mammals, especially bats and crickets. We have less of an understanding of how bats are listening to crickets, but we know they are, because you can, people who work on bat-cricket interactions have done these really cool experiments where they leave a bat in a room and then they leave a cricket ko aise dhage se bandh ke, like they just leave the cricket hanging. And of course the lights are off, and the bats are able to find and hunt the crickets in complete darkness. Now, in India, most of our bats are not just using ultrasound. They're also using movement sounds of the cricket and so on. But through they don't need light at all in order to hunt these crickets. So they're very good at finding them. And as soon as these crickets call, in fact, you can, instead of putting the cricket, you can just put a speaker and they will try to hunt the speaker. So they are clearly using the cricket calls to find them. So we think that these 4 to 5 call range, it's not because the crickets are choosing ki main aaj is frequency range use karunga, etc. These are hardwired calls. They don't have any control. One of the reasons we're looking at insects is because unlike birds and us, they're not consciously changing the way they're producing sound. But the sound, there's one sound that they produce lifelong. But basically the animals who produce calls, which other animals can hunt well, they are being eaten and they're not surviving. And the ones that survive are the ones who happen to produce calls in these frequency ranges. So it's a very interesting story. You've asked actually all the perfect questions that tell us more about, that enable me to talk more about why this is such a cool system. But yes, lots of examples of coevolution.
Thank you. I have a lot of questions, but I will give to others.
Please feel free.
Thank you.
Yeah.
Any other questions?
I have one short question. Can we use this sound frequency to controlling this pest?
Say that again. Can we use this sound control frequency too?
Hello.
Can you this question? Can we use the sound frequency too? To control something.
To control the what is that? The cricket.
Oh, the pest.
Pest.
Yeah. In theory, you can. In theory, you can produce sounds which are attractive to the cricket, or you can make bad sounds. So perhaps the least unfriendly thing you can do to these animals is you just put up a speaker that emits bat sounds. And in theory, these animals should not want to be near bats. But in practice, they are around bats. If they were to completely avoid bats, they would not be able to live their lives. So, but in theory, yes, any kind of advertisement that you have predators that eat these animals, whether it's olfactory, so those long antennae are used to smell. So if you have lots of dog and cat pee on your farm where pests are coming in, and or you use acoustics to scare away the animals from fields, both of these should in theory work to help with pest control. And the major pests, the ones that eat lots of food, are not our night wala crickets and bush crickets. They're mostly those locusts and grasshoppers, which I showed you in the first slide. But yeah, same principle.
Thank you, Dr. Sanan. Any other have any questions? So you can ask. We have 3, 4, 5 minutes more.
Thank you, Dr. Rajaraman, for your enlightening lecture.
My pleasure.
Yeah, actually, probably I missed something, but I was actually working. But I just want to know the reason. My little question is, I just want to know the reason, because in ecology, probably I missed something from your lecture. In ecology, we studied that all these things that are happening that you just mentioned are a part of courtship behavior.
Right.
So is that true? And my second question is about I don't know what it is called, that firefly. Yes, the firefly.
Right. Excellent. Yeah.
It is actually on the verge of extinction, because I don't think it is able to find some another way to adapt without the light for its courtship behavior.
That's right.
So do you have anything regarding the same?
Right.
Because our childhood is gone
Exactly.
with them only at night.
Wahi to, exactly, correct. Yeah, both very, very good questions. Actually, you didn't miss anything. In the sense, your questions are both excellent questions. So answer the first one. So there's technically these are also for mating, but they are the difference between this is what's called a mating advertisement call. Like you're sending out an advertisement into the world कि मैं यहाँ पे हूँ. If anyone wants to mate with me, here's where you can find me. But a courtship call is technically defined as the call you make when you know that there's a mate nearby. So you're courting, matlab you're making an overture. So these animals don't always call the exact same way when another animal is around. This, what I've shown you so far, is what's called the advertisement call, which is agar ye animal ekdam akela hai to bhi, come 5 o'clock in the evening, if they're in an akela in a dabba in the lab, they'll still call this call. And then if you give them a mate, then the courtship part of the call begins, which is a little more interactive sometimes. It may change also. They often, when there's a mate actually that they have spotted nearby, either visually or they've smelt olfactorily, they have noticed it, then often they produce more of the call. So if you don't know that there's a mate nearby, then they just produce the call sometimes. But when the mate is around, they are more persistent. So the courtship call is sometimes different from this. This is the advertisement call, which is being heard by everybody. It's being heard by your predators and it's being heard by your, conspecifics. Second question that you asked was about fireflies, right? And in fact, we share a lot with the firefly community because fireflies also produce its visual signals, but everything else is the same. There's a patterning of the blinking of the light. Then Mycopoda coordinates the calling between different animals. And so do fireflies. You can have coordinated fireflies. In Western Ghats, you have these huge groups of coordinated fireflies. But we grew up with fireflies everywhere. And now it's so hard to find fireflies. I still take people to 2 or 3 urban forests of Delhi, just to where Aravalis are still, undestroyed, where you can, mushkil say, you'll see 5 or 6 of them, sometimes 10 of them, but not the huge numbers that we used to see earlier. And yes, light pollution affects them a lot. And many other things really hugely affect them. And likewise, sound pollution affects our crickets. The only difference being that crickets are so loud. As I said, they're 100 dB at source. So sometimes they will outcompete concerts also. And, all the different sounds, horns and so on that we have at night. We're mostly reducing their numbers through destroying their habitat. So these crickets, bush crickets, to lay their eggs on vegetation. So every time we cut vegetation, I have been trying to get my university to not trim the hedges and shrubs and all, because usi mein ande honge. So, but yeah, there's no question that they're on they are reducing in number. There are very few insects which are placed on an extinction watch list because, as we like to say, charismatic species, mostly big mammals, are the ones that, and some birds tend to be on these lists. but we have so few estimates of the baseline insect diversity in India, let alone how it's changing over time, that we are able to anecdotally see and hear the difference. There are places where we used to hear animals 10 years ago, we don't hear them now. But we need to build those rigorous kind of data sets. And so one of the things we're proposing is to use bioacoustics, that is just recording the environment on a regular basis as a way to get some kind of non-invasive estimate of biodiversity on a regular basis, so that we can actually have data to say कि 10 साल पहले ये था and अब नहीं है इस area में या उस area में. So yeah.
Is there any way to save them?
I think it's the same as with many animals. It's just preserve more of the habitat as it is. If we need to build more buildings, that's fine, but let's build upwards, not sideways so much. These are some of the things which we will just have to do going forward, I think, so we can conserve them. Yeah.
Thank you.
Thank you. Both all excellent questions. I really enjoyed the Q&A session.
Thank you. Is there any other question? Dr. Bittu, just want to introduce Professor Balram Singh sir. He is the President and Director of Institute of Advanced Sciences, USA, and here in KSAS. Probably it's very early in the morning in US, so I think sir is preparing because sir is here.
I'm sorry. I really enjoyed. I wonder, Rajkumar didn't ask this question. I wonder if we ever use botulinum toxin, neurotoxin, to see if that will work in the kind of nervous system that you were talking about.
Oh, yeah, they will. They absolutely will. In fact, they've used pharmacology to figure out in that cricket system, it looks like there are some, I'm trying to remember what they used. So in order to, of course, it's insects, so it's a mostly, so in fact, the drugs that produce the strongest kind of effects are acetylcholine, pathway inhibitors. And these, in fact, we also have data from the lab that I can show you where if you inject these into the brain, then that actually seems to remove the inhibition of the call production neuron. And so the animal will call in the dish, even if it doesn't want to, right? If you use drugs. So yes, pharmacology is very helpful on these animals. We tried to do the least minimal thing to the animals, which is to just snip the cord and then let the animal recover, and then it fully regains its call function. But when the animals, don't call at all, then you can finally induce the call by injecting a small amount of acetylcholine pathway, agonists in the brain.
Well, Rajkumar, now we have another animal model to try to fix people's speeches. That is wonderful. I'm sorry. We are continuing with our botulinum research conference,
Right.
which is started yesterday in the evening, and I will be running down there in about half an hour. And
thank you very much for attending, all of you.
Yeah, but it's wonderful. It's wonderful. Yeah. We got some good ideas there.
Thank you very much.
Thank you. Sure.
Thank you, Dr. Bittu, and thanks all for joining this KSAS talk this month. So next month we will come with another speaker, another topic, and hope you all enjoyed it. Thank you. Thank you.
Thank you very much. Thank you so much.
Thank you.